hope my voice is audible. So, a very good evening to all of you. Today we'll start the week 3 lectures. So, in week 3 we have covered about RNA transcription, and in week 3 there are total 5 lectures. So, one by one we'll start the revision of each lecture, and then we'll try to solve some questions related to that lecture. And in the end, if you have any questions, any queries, you can ask me. Okay, so we'll start now. So, the lecture 1 in week 3 is about RNA transcription, the central dogma. So, when we talk about the central dogma, what does it mean? So, it says that DNA, it first replicates itself. Just a minute, I'll have the pointer. Yeah. So, as you can see that this is the DNA. Okay, so DNA replicates itself with the help of an enzyme which is known as the DNA polymerase. So DNA, it makes copies of itself with the help of DNA polymerase. Now this DNA, it can be converted into RNA with the help of an enzyme, DNA dependent RNA polymerase. This RNA can be replicated with the help of RNA dependent RNA polymerase, and eventually the RNA is converted into proteins. Now, there are many important points to note over here. First is that so when DNA is converted into RNA, it is the positive strand, that is the plus strand RNA, which is formed first. And when this RNA replicates itself, then the minus strand RNA is formed. So always remember that plus sense RNA is formed first during transcription. It is very important. Now this RNA, it can be converted back to DNA with the help of the enzyme reverse transcriptase. So DNA forms a copy of itself. DNA to DNA is replication with the help of enzyme DNA polymerase. DNA to RNA with the help of RNA polymerase. RNA to DNA with the help of enzyme reverse transcriptase. So this whole uh, cycle, or this whole process, is known as the central dogma, right? Now, in this central dogma, there are 4 important enzymes. The first one is DNA dependent DNA polymerase. This DNA dependent DNA polymerase is required here for the replication of DNA. Then there are 2 RNA polymerase, and here also RNA polymerase. But what is the difference in the functioning of these 2 RNA polymerase? What can be the difference? What is the difference in the functioning of these 2 RNA polymerase? So these both are RNA polymerase only, but the first one is DNA dependent, the second one is RNA dependent. What does it mean? What does it mean? It means that for the transcription of DNA into RNA, the polymerase that is needed is DNA dependent. But for the replication of RNA, the polymerase that is required is RNA dependent. So first one is your DNA dependent RNA polymerase, and the second one is your RNA dependent RNA polymerase. Okay, so this is about the polymerase. Then the fourth enzyme is reverse transcriptase. So transcription, if I say transcription is the formation of RNA from DNA. DNA is converted into RNA. This is your transcription. If RNA is converted back into DNA, what would it be called? The reverse of transcription. So it would be reverse transcription, right? So the enzyme which is required for this reverse transcription is reverse transcriptase. Ase means enzyme, the enzyme which is causing reverse transcription, that is reverse transcriptase. Okay, Now, some important points regarding the central dogma is the two strands of DNA are not identical to each other, they are complementary to each other. What does it mean? So just a minute. I'll add a new slide, okay, and I'll write a DNA sequence. Suppose the sequence is AA TT CCGTA. Okay. Now, what is the complementary of this sequence? What would be the complementary sequence? The complementary sequence would be T T A A G G C A T. This is the complementary sequence. And if I write only this A A T T G G G T A, then this is your identical sequence. So what they are saying is that the two strands of DNA are complementary and not identical. That means in DNA, if the sequence of one strand of DNA is this, the sequence of other strand of DNA would be this, because the two strands of DNA are joined together by hydrogen bonding. So hydrogen bonding can be formed between A and T and G and C. Between A and between T and T and between C and C and between G and G is not, there is no hydrogen bonding. So in order for the two strands to remain together, there has to be hydrogen bonds. And hydrogen bonds can be formed only when the two strands are complementary to each other. Therefore, it is important to always remember that the two strands of DNA are not identical to each other, they are complementary to each other. Fine. Next point is retroviruses have RNA genome. So we have studied that although uh, through the evolution, the course of time, the genome is now DNA. DNA has evolved as the genetic content, genetic material of the organisms, but certain viruses still have RNA as their genome. For example, retroviruses. So retroviruses have RNA as their genomes. So can you give me an example of a retrovirus which has RNA genome and which has really caused havoc in recent times? Any example that anybody can think of? Okay, so the example for this is coronavirus. Okay, the COVID is a disease, the virus is coronavirus. So the coronavirus is a retrovirus that has RNA as the genome. Okay, and because it has RNA as a genome, so what are the different properties because of which DNA was evolved as a genetic material? Because DNA was stable. There is proofreading activity that happens when DNA replicates. But when RNA replicates, there is no proofreading activity that happens. What is the meaning of proofreading activity and how does it actually helps? Suppose if you have a DNA and it has to incorporate A, T, T, C. Now it has to make a strand, it has to incorporate T, T, A, and here also T, but by mistake it has incorporated a C. Okay, now this is a mismatch. So while proofreading, during DNA replication, they will see this mismatch and they will try to correct it. So this mismatch would be repaired, and if there are chances that some incorrections cannot be repaired, then the DNA replication is halted. So what happens, this mutation is not passed to the generations, but in the case of RNA, what happens that the mismatch is not repaired, so that mutation is passed on. So RNA genome always evolve, and that is the reason why in COVID we have so many different variants of the virus, because the virus has RNA as their genome, and RNA is constantly mutating and changing its genetic content. And that is the reason why it is very difficult to make a single universal vaccine for RNA for COVID viruses, because RNA is constantly evolving, its genome is mutating. So that is the point. Retroviruses have RNA as their genomes, and these are the stop codons, okay, and these are the sense codons. So when we talk about codons, so codons, they can either be sense codons, okay, or they can be stop codons. Sense codons means that they make some sense, they are making some sense, and in the respect of molecular biology, that sense is making an amino acids, okay. So when I say sense codons, that means these are the codons that make an amino acid, that codes for an amino acids, whereas stop codons give signal so that the translation is terminated, translation is stopped. So sense codon means that the codons are making some sense, and with respect to molecular biology, that sense is making an amino acid, okay. So how many codons are required for to code one amino acid? How many codons are required? 3 codons. So 3 codons, they code for one amino acid, right? Okay, so this was the last point, sense codons, where stop codons are UAA, UAG, UGA, and sense codon when an amino acid comes in. Fine. Now we come to the next part of this chapter, that is genotype and phenotype. So what is the difference between genotype and phenotype? Genotype describes the genetic content of an individual, genetic content, and phenotype is the expression of that genetic content. For example, if we talk about our genetic content, so what the genes, all the genes are present everywhere. For example, if I talk about lungs, okay, and I am talking about eyes, so all the genes that are required in specifically in lungs, they are also present in the DNA of eyes and vice versa, but they are not expressed because lung specific genes are only expressed in lung area, whereas eye specific genes are expressed only in the eye region. So what does it means? That although all the genes are present in both lungs and eye, the expression is cell specific. You should always remember, this is a very important term, that there is cell specific expression, okay. So we have cell specific expression. So some genes are expressed only in some particular cells, but that doesn't mean that these genes are not present in other cells. They are present, but their expression is not there. So if I say, if I have someone has a darker skin tone or lighter skin tone, or height, short height, long height, or the color, anything, any phenotypic observable characteristic, that is not only because of the presence or absence of the gene, it is more on the expression of the gene. For example, here you can see in this figure, genotype is an organism's genetic information and phenotype is the set of observable physical traits, okay. And for example, you see this is purple. Why it is purple? Because the genotypic content is BB. So capital B, capital B, they are both homozygous and they both are dominant. Even if one is present, it would be purple because capital B is there, it is dominant. Here both recessive are there, so it is white. Just an example to show how genotype and phenotype varies, okay. So in this whole this thing chapter lectures for this week, so I have tried to uh, uh, take some of the images that I could sense are important with respect to your examination or in general from the from sir's lecture as well. So I have written in the slides, if you can see that from where they are taken. If some images are taken from the lectures, then I have noted it there, so that you can just go through them if you have any problems, and then you can ask me. Fine. So now this is genotype versus phenotype that we have studied. Now next thing is transcription and translation in prokaryotes and eukaryotes. So now the process of transcription is different in the case of prokaryotes, and the basic concept, the basic reason and the logic, that is the same, okay. The logic remains the same, but there are certain differences in the transcription process between prokaryotes and eukaryotes. So can you tell me why there are some differences and what is the basic reason of this difference? Why transcription and translation are different in prokaryotes and eukaryotes? What you can think of a possible reason behind this? Think. you see, the difference lies in the genetic makeup of the cell of a prokaryote and a eukaryote. So prokaryote, as we know, it does not have a defined nucleus, okay, whereas eukaryotes, they do have a defined nucleus. Now what does it mean? That means whatever is happening in a prokaryotic cell, it is happening in the same cytoplasmic area, whereas now here you have boundaries, so things can be done in different places. So if you just look at a prokaryotic cell, I'll try to maximize it for your convenience. Okay, just a minute. uh, yeah. Hmm. Now you see it here, you can see that this is the cytoplasmic area, right? And this is the DNA which is dispersed in the cytoplasm. This DNA is not present in a specific nucleus in the case of a prokaryotic cell. So this is your DNA and this is the mRNA. So this mRNA is getting formed from DNA, and you can see that these are the ribosomes which are attached to the mRNA, and this is your protein. So what you can observe from here is that transcription and translation are occurring simultaneously. There is a simultaneous transcription and translation happening in the case of a prokaryotic cell. Now switching to eukaryotes, you can see this is the DNA. It is present in a well defined nucleus, right? And in this well defined nucleus, this is the DNA which forms first the pre-mRNA. What is this pre-mRNA? It is the premature mRNA which is not completely processed. So this pre-mRNA is first processed within the nucleus, and it gets converted into the mature mRNA, right? Now this mature mRNA, it comes out of the nucleus into the cytoplasm, and in the cytoplasm translation happens. So first mRNA is formed, it is matured, and then it comes out of the nucleus, and then translation takes place. So unlike in the prokaryotic cells where transcription and translation was happening simultaneously, in the case of eukaryotes, transcription and translation does not occur simultaneously. First transcription happens in the nucleus, and then the primary transcript is converted into a mature mRNA that comes out of the nucleus, and in the cytoplasm translation happens. So this is one of the major differences between the transcription and translation in the case of prokaryotes and eukaryotes. Another major important difference is, you can see over here, is that in the case of eukaryotes, as we just discussed, that the mRNA that is formed after transcription is a primary transcript. It has to undergo some kinds of modifications to become a mature transcript. And what are those RNA processing events that takes place? So essentially there are three important events that takes place. First one is splicing of the intronic regions. So you can see these are the intronic regions. Okay, this is the white portions are the intronic regions, and you can see with the help of splicing, those intronic regions are removed. So the first kind of modification that occurs in the case of eukaryotes is splicing. Second is your capping, 5 prime capping that takes place. Now what is this 5 prime capping? The 5 prime end is protected with the help of the addition of some 5 uh, we'll see 5 methyl guanosine, and this protects the mRNA from exonuclease activities. This is the second kind of modification that occurs. The third kind of modification that occurs, you can see on the 3 prime end, is a poly A tail. So there is a poly A tail, polyadenosine tail, which is added in the 3 prime end. So these are the three major RNA processing events that takes place, especially in the case of eukaryotes, and it does not occurs in the case of prokaryotes. So these are the two major differences. Uh, of course, there are many more differences, but these two differences are the major reasons why all other differences takes place. Now there is one question that comes, that why does uh, um, transcription and translation occur simultaneously in the case of prokaryotic cell? Why it does not happen that, okay, fine, that there is no defined nucleus, understandable. But then within the nucleus only, first they can complete the transcription process and then they can start the translation process. Why to hurry? Why does the transcription and translation happens at the same time? Is there any reason behind it? Because there must be a reason. Think of a reason. Well, the reason is that so you can see, if you know that the cytoplasm has a reducing environment. Okay, it has a reducing environment. So that means in the cytoplasm, RNA is very much prone to degradation. So if transcription and translation are not occurring simultaneously, then as the RNA is formed, and if we just wait for the transcription to complete and then this RNA translation to happen, then by that time the translation starts, RNA might have already degraded. So when as soon as mRNA is forming, continuously ribosomes are getting attached to it. So this attachment of the ribosomes in a way also prevents the mRNA from degradation. Okay, and because since there is no well defined nucleus, the mRNA does not have to come out, then why to waste time and wait for the full mRNA to be formed to start the transcription process? So therefore it occurs simultaneously. So these are the two reasons. Okay, then comes types of RNA molecules. So there are various types of RNA molecules like mRNA, tRNA, rRNA. mRNA is the intermediate that carry the genetic information from the DNA to the ribosomes, that is, it is involved in translation process. They all are involved in transcription and translation process. tRNAs are adapters between amino acids and codons. Ribosomal RNA are the structural and catalytic components of ribosomes. So these are the RNA molecules. There are some other kinds of RNA molecules. These are non-coding RNAs. They both are non-coding RNAs. Coding RNAs means RNAs which codes for an amino acid. Non-coding RNAs are the RNA that does not code for an amino acid. Okay. These are small nuclear RNAs, microRNA. RNA interference is a process that we'll study eventually. And then there are PV interacting RNA. These are all non-coding RNAs. Okay, so these are the types of RNA molecules. So now we have completed our lecture number 1. So in lecture number 1, just a quick revision, we have studied about the central dogma and the 4 important enzymes. Then we have studied the difference between genotype and phenotype. Then we have seen the difference of transcription and translation in the case of prokaryotes and eukaryotes. And then lastly, we saw the types of RNA molecules and various non-coding RNA molecules, etc. Now we come to the next lecture. I'll try to do it in a full screen. Yes. So lecture number 2, which is RNA transcription initial steps. So now what I'll try to so in my PPT I have tried to make slides in a way that this RNA transcription is very clear to you. So I have very specifically divided the 3 steps in 3 different slides, and then you can just go through them and get an idea of what RNA transcription is, what are the different processes of RNA transcription. So this is, you can see just a general overview of the RNA transcription process. This is the double-stranded DNA. Now, one important difference between the replication and transcription is that in replication, both the DNA strands are involved in the replication process, whereas in the case of RNA transcription, only one of the strand is responsible for the transcription process. Okay, suppose this is your DNA, this is double-stranded, right? Now, both the strands are separated. Now both the strands will act as a template for the formation of DNA molecules. This is replication. Okay, both the strands are involved. Here both the strands act as template. Replication, both the strands acts as template. If I talk about transcription, this is your DNA molecule. Okay, it is broken down and only one of the strand is responsible for the formation of the RNA. Okay, so here in transcription, what is happening? Only one DNA strand acts as a template. Is it clear? Now, RNA synthesis, single strand of DNA is acting as a template. I just told you, you required ribonucleoside triphosphates, okay, and there is no pre-existing primers. So what is the meaning of this? For the DNA replication, you need some primers to start, whereas for RNA, you need not to have any primers because RNA has its own catalytic function. This is like a general that just a general overview. Now the transcription bubble is an important thing. You see the DNA is double stranded, okay, and only one of the strand acts as a template. So what happens that for the transcription to take place, the DNA is first melted and the two strands are separated, and when the two strands separate, it kinds of looks like a bubble, therefore it is known as a transcription bubble. And when the two strands are separated, then the RNA polymerase comes and bind to it, and then RNA is formed. You can see this is the image I have taken from Sir's class only. You can see RNA polymerase binds specific nucleotide sequence. So this is your DNA. It is melted, temporary melting is there, and this temporary melting causes the RNA polymerase, this is the RNA polymerase, it binds, and then your RNA is formed. Now important thing to note over here is that RNA polymerase that binds, within this melted region there are certain specific locations where RNA polymerase comes and binds. So RNA polymerase does not come like just comes and bind anywhere. No, it has certain specific sequences, nucleotide sequences, which are known as the promoter region sequences, where the RNA polymerase binds. And here we are talking about eukaryotic transcription. So in eukaryotic transcription there is single RNA polymerase, fine. Then this winding, the unwinding that happens is mostly the AT rich region where unwinding is happening, okay. So unwinding mostly happens in the AT rich region because AT rich has only two hydrogen bonds, it is easily to unwind that region. Now, so these are some of the general features of RNA synthesis. So RNA synthesis is similar to DNA synthesis except the following points and some others that you will find in your lecture. The precursors are ribonucleoside triphosphate, not deoxyribonucleoside triphosphate as in the case of DNA. Next point is only one strand of DNA is used as a template. We have just discussed this point, that in the case of replication both the strands are used as a template, but in the case of RNA transcription only one of the strand is used as a template. Third point is RNA chains can be initiated de novo because RNA has catalytic functions. RNA has catalytic functions, so it does not require it does not require primer, so it can be initiated de novo. Whereas in the case of DNA you need some kinds of primers for the initiation of the reaction. The RNA molecule will be complementary to the DNA template. So if this is your DNA, sense antisense. So this antisense is the strand which acts as a template. So RNA would be complementary to the antisense strand. Okay, now because RNA is complementary to the antisense strand and sense strand is also complementary to antisense strand, so RNA is actually identical to the sense strand, which is the non-template. Template strand is known as antisense strand. Remember, template strand is the antisense strand, and non-template strand is the sense strand. Is it clear? Okay. Then RNA synthesis is catalyzed by RNA polymerase and it proceeds in the direction of 5' to 3'. Then for the opening of the transcription bubble, so the transcription bubble that opens, it opens at the AT rich region, and the region in prokaryotes it is known as ORIC, and in the case of eukaryotes it is known as autonomously replicating sequence, but this is also an AT rich region. Fine, now we'll do some questions. The first question is, how many DNAs, this question we have just discussed, how many DNA strands act as a template during transcription and replication? Both the strands, only one respectively, one or both the strands respectively, one and one, both the strands in both the cases. So what is the answer? Now just a minute. Yes. Tell me, what is the answer of this question? How many DNA strands act as a template? So during transcription, only one strand is needed, whereas during replication both the strands are needed. So the answer would be B. Fine. Next question is, which strand act as a template during transcription? We have just discussed this. Sense strand, antisense strand, any strand can act as a template or none of the above. So what is the correct answer? Just a minute. So antisense strand is the strand that acts as a template during transcription. Next question. How many amino acids are formed if we have 21 bases available? So we have how many bases? Number of bases is 21. And how many amino acid? For one amino acid, we need 3 bases. So therefore, it would be 21 divided by 3. 7 is the answer. Okay, 7. Next question is, replication and transcription are both bidirectional, both unidirectional, bidirectional and unidirectional respectively, unidirectional and bidirectional respectively. What is the answer? Replication and transcription are what? So the answer is, replication is bidirectional and transcription is unidirectional. Why you think it is like this? Why replication is bidirectional and transcription is unidirectional? The answer lies in the number of strands that are used as a template. Think about it, and then we'll also see. Think what can be the reason. Okay. So, if we talk about replication, all right. So in replication, this is your strand 1, this is your double strand. Now you have 2 strands. Okay. This is 5' to 3 prime, 3 prime to 5 prime. Now you see here, primers are attached at the 3 prime end. So for this strand, this is the direction. For this strand, this is the direction. Is it not so? Yes or no? Because primers are attached at the 3 prime end and it has to go in the direction of 5 prime to 3 prime. Replication, any nucleotide, so this is replication or RNA transcription, it happens in the direction of 5 prime to 3 prime. 5 prime to 3 prime. So in both the directions, in replication, the synthesis is happening. Okay, and it also has to do with the leading and the lagging strand concept, if you know the concept. Fine. Now I am talking about transcription here. Okay, now I come to transcription. Okay, this is the DNA. Now this is your double stranded DNA. This strand is only acting as a template molecule, because in the case of transcription, only one strand acts as a template. This is, if it is 5 prime to 3 prime, this is the direction. There is no other direction because only one strand is acting as a template. Therefore replication is bidirectional, whereas transcription is unidirectional. I hope it is clear to you. Now, so with this we have completed the lecture 2. So in lecture 2 what we have studied? So in lecture 2 we have first studied like just a basic general introduction about the RNA transcription. We have studied that how only one strand of DNA is responsible for the synthesis of RNA, and then we studied about the transcription bubble, that how the DNA is locally unwinded, and in that locally unwinded region the RNA polymerase comes and it binds to specific nucleotide sequence within the unwinded region, and those nucleotide sequences are actually the promoter regions, and then your formation of mRNA takes place. Then we have studied about some basic features of RNA synthesis. What is a sense strand, what is the antisense strand, what is a template strand, and what is a non-template strand. Then we have solved some questions and we have studied so this is about the initial introduction. So in the first lecture we have studied about the central dogma. Second lecture we have studied about the initial description. Now we'll see one by one the various different stages of transcription. So again, some general features about the transcription process. Just a minute. Yeah. Okay. So you can see some general features. It requires a DNA dependent RNA polymerase. It is very important to remember DNA dependent RNA polymerase. RNA dependent RNA polymerase is needed during replication of RNA, but during transcription you need DNA dependent. Fine. Synthesis occurs unidirectional in 5 prime to 3 prime direction. Unwinding of the DNA template occurs to form it forms a transcription bubble. And essentially there are three main steps in the transcription process: initiation, elongation, and termination. So these three steps remains same for, the same three steps are in the case of prokaryotes and eukaryotes, but there are some differences. So first we're talking about the prokaryotic RNA transcription. So in prokaryotic RNA transcription, so whenever we're talking about transcription, as the steps are very important, similarly studying about the RNA polymerase is equally important or sometimes even more important. So the RNA polymerase is different in the case of prokaryotes and in the case of eukaryotes. In the case of prokaryotes, you just have a single RNA polymerase, but in the case of eukaryotes, you have different types of RNA polymerase 1, 2, 3, 4, 5. So RNA polymerase in general is made up of different subunits. Different subunits combine to make one RNA polymerase. So the number of subunits that combine to make a prokaryotic RNA polymerase is different from the number of subunits that are required to make a eukaryotic RNA polymerase. That means in prokaryotic RNA polymerase, the number of subunits are lesser in comparison to a eukaryotic RNA polymerase. So eukaryotic RNA polymerase has greater number of subunits. It is a bigger and a more complex RNA polymerase, and there are different kinds of RNA polymerase. Whereas in the case of prokaryotes, there is only single type of RNA polymerase, and there is lesser number of subunits are there. This is like a basic general difference of the RNA polymerase of the prokaryotic cells and eukaryotic cells in the transcription process. So you can see this is an important slide. Okay, this is an important slide not just for your NPTEL examination point of view, but for your other competitive examination per se also. So you can see that the E. coli RNA polymerase, it's a tetrameric core. So whenever we talk about a polymerase, there are two things. First is a core enzyme, and the other one is holoenzyme. Okay, core enzyme which has two alpha subunit, beta dash subunits, and in the core enzyme when you add the sigma factor, then it becomes your holoenzyme. Okay, now the functions of the subunits, again important topic. So what is the function of alpha subunit? It is required for the assembly of the core polymerase. Always remember, alpha subunit is responsible for the assembly of the core polymerase. It is not required for the assembly of the holoenzyme, core enzyme. Then beta subunit is responsible for the ribonucleoside triphosphate binding site. Beta dash is required is a DNA template binding region, and sigma factor is required for the initiation of transcription. So what does it mean? What does this mean? Sigma factor is very important, guys. Why sigma factor is important? These are your different RNA polymerase subunits. Okay, I'm not counting the number of subunits here. This is your DNA template. This is the promoter region. This is the promoter region, right? Now in this promoter region, this RNA polymerase has to come and bind. Now it cannot just come randomly, and it cannot just sit anywhere in the promoter. It has to bind at a specific location, and there only it has to sit. So how it will recognize that specific location? This specific location is recognized with the help of sigma factor. So the sigma factor of the RNA polymerase is actually responsible for the initiation of transcription, because it is coming and it is sitting over the promoter region. It recognizes the promoter region. So this is important to understand, right? So the different subunits have different functions. Alpha subunit is responsible for the assembly of the core complex. That means the four things, tetrameric core. Beta is responsible for the ribonucleoside triphosphate binding site, beta dash DNA template binding region, and sigma factor initiation of transcription. So now we come to transcription initiation. So the first event in transcription occurs, the initiation of transcription. So how the transcription is initiated? Like a story, you have to, if you're reading it like a story, it would be very easy for you to understand. This is your DNA template. Okay, so the things that I have written in bold are the important points. Okay, see, this is your polymerase, RNA polymerase. So RNA polymerase holoenzyme, that means sigma factor is also there. So RNA polymerase holoenzyme, this is the sigma factor, binds to the promoter region of the DNA. Recognition of the promoter occurs by sigma factor. This is the DNA template. RNA polymerase will come, and then sigma factor will recognize the promoter site, and then RNA polymerase will bind to it. This is known as closed binary complex. Why it is known as closed? Because up till now the DNA has not melted and DNA is not opened, so it is closed. Binary complex because they are just two things, DNA and RNA polymerase. So it's a closed binary complex. Then what happens? So first thing is that the holoenzyme comes and it binds. Okay. Second thing is unwinding of the DNA strand takes place to provide a single stranded template. Now what happens? The melting of the DNA happens. So now you can see that there is a region where the DNA melting is taking place. So this is the unwinding of the DNA. Now it is an open binary complex. It is an open because now the DNA has melted, but still it is binary because there are just two things, RNA polymerase and DNA template. So unwinding occurs. Okay, fine. This has happened. This has happened. Third is formation of the phosphodiester bond. Then you can see formation of the first phosphodiester bond takes place. After the formation of a first phosphodiester bond, there is a small mRNA, small RNA that has formed. And now it is known as a ternary complex. Why ternary complex? Because there is RNA polymerase, DNA template and small mRNA that has formed. So this is a ternary complex. Then the sigma factor is released and the elongation takes place. So why you think the sigma factor is released? The sigma factor is released because now the process has completed. Now there is no use of sigma factor because the main point of sigma factor is to recognize the promoter region. The promoter region is recognized, now sigma factor is not needed. But why does the RNA polymerase has to release the sigma factor? It can be bounded to it. It can continue its work. Why it has to release the factor? Can you think of a possible explanation? Why it has to release the sigma factor? Why it cannot continue with the sigma factor? Think. Can you think of a possible explanation? Why it has to release? Okay. So the reason why the sigma factor has to be released is because the binding of the sigma factor to the RNA polymerase is an energetically expensive process. That means a large amount of energy is required to keep the sigma factor binded to the RNA polymerase enzyme. But since there is no need of RNA polymerase now, then why to just for no use expend that much of energy? So that is why the sigma factor is released. For example, even in our body, so we say some, like for example tailbone, so there is no tailbone, we have a tailbone but there is no tail here. Why the tail during the course of evolution was completely vanished? Because there is no use of it. So unnecessary organs, they are eventually in the course of evolution they are just wiped off because to keep them is requires energy, so that would need a lot of energy. So if you do not need something, then there is no need of keeping it in the body. Likewise here also you can see that since the function of sigma factor is just the recognition of the promoters region, it has completed its function. Now it is no longer needed, and because it is no longer needed, it can be removed because it is an energetically expensive process to keep the sigma factor remain with the RNA polymerase. And that is the reason why after the formation of the first phosphodiester bond, the sigma factor is released, and then something which is known as promoter clearance takes place, and then eventually the transcription initiation is completed. So transcription initiation completes with the formation of the first phosphodiester bond. As soon as the first phosphodiester bond is formed, the transcription initiation is completed, and then what happens is that the transcription bubble, it keeps on moving on the DNA template. It looks like that the transcription bubble is walking on the template, okay, and then the phosphodiester bonds are formed and mRNA is lengthened. Now next we come to the E. coli promoter region. So as we talked about that the RNA polymerase sigma factor subunit, within the transcription bubble it binds to specific regions. It cannot just bind anywhere, and those regions are actually the consensus sequences. Consensus sequences means sequences which are highly conserved, sequences which are highly conserved. So these are the consensus sequences where typically the sigma factor comes and binds. So there are two important consensus sequences. The first is -10 sequence, another one is -35 sequence. Tata at and TTGACA. So the RNA polymerase comes and it binds to -10 region and -35 regions only. And the transcription start site is +1. Okay, now I have to just explain you one thing before we move further. What is the meaning of this -10 and -35? Can anybody explain it? Think of a reason. What is the meaning of this -10 -35? What does it signifies? What does the minus sign here signifies? We are not doing any mathematical calculations here. What does this minus sign signifies with respect to molecular biology here? Think. I'll give you 1 minute to think. Okay. So this is, if this is your DNA template, this is your downstream, upstream. Fine. So here you can see that this is the +1. So transcription occurs, the site at where transcription occurs, the transcription start site is denoted as +1. Okay, so transcription start site is denoted as +1, and anything which is before this has minus, like -2, -3, -4, and it comes to -10, and it goes to -35. So the sequences at -10 and -35 are the regions where promoter regions where the RNA polymerase comes and binds, and downstream are like +2, +3, +4. So this site, which is the left of the transcription start site, are known as upstream sequences, and this side, which is the right side of the transcription start site, are known as downstream sequences. Okay, so is this clear what I'm trying to tell over here? So that is why -10 -35. So the two consensus sequences with where the RNA polymerase comes and binds, first is -10, the other one is -35. Is that clear to everybody or not? It's not clear, you can ask right away. Fine. So okay, so we Okay, quickly we'll see these questions. Sigma factor is important for RNA transcription termination, recognition of DNA regulatory region, movement of holoenzyme, and RNA transcription initiation. What is the correct answer? Sigma factor is needed for what? Transcription termination, recognition of the DNA regulatory region, movement of the holoenzymes, or RNA transcription initiation. We have just studied about it. So the correct answer is RNA transcription initiation. Okay, sigma factor is responsible. Sigma factor is responsible for the RNA transcription initiation, because sigma factor is actually responsible for the now. Next is assembly of the core complex occurs with the help of alpha subunit, beta dash subunit, beta subunit, or gamma subunit. Remember, we are talking about the core complex, not the holoenzyme. So what is a core complex? Core complex has alpha 2, beta, beta dash, and then plus the sigma factor, it becomes holoenzyme. So the assembly of the core complex means this tetrameric core. So the assembly of the core complex occurs with the help of alpha subunit, beta dash subunit, beta subunit, or gamma subunit. The correct answer is alpha subunit. So just a minute. Yeah. So in lecture 3, before moving to elongation, we'll just revise. So these are the general features which requires DNA dependent RNA polymerase. Synthesis occurs in the direction of 5' to 3'. Then unwinding of the DNA template occurs, which ultimately forms a transcription bubble. And then the three main steps are initiation, elongation, and termination. Then there are the RNA polymerase in the case of prokaryotes and in the case of eukaryotes are different. In the case of prokaryotes, it is a simple RNA polymerase and just one RNA polymerase is there. In the case of eukaryotes, there are 5 different kinds of RNA polymerase 1, 2, 3, 4, 5. So you can see that the E. coli RNA polymerase has a tetrameric core, a core complex, and a holoenzyme. So core has two alpha subunits, beta and beta dash. Holoenzyme has alpha 2, beta, beta dash, and sigma. Then what is the function of the subunits? Assembly of the tetrameric core is done by alpha subunit. Beta subunit is responsible for ribonucleoside triphosphate binding site. Beta dash is responsible for DNA template binding region. And sigma factor is most important because it is required for the initiation of transcription, which means that the promoter is recognized with the help of sigma factor. You can see here E. coli RNA polymerase core enzyme, what I have written, 2 alpha subunits, beta, beta dash, and holoenzymes. It requires additional sigma factor, which is responsible for the initiation of transcription. And then we have studied about the initiation process. Initiation requires essentially 3 major points. First is the binding of the RNA polymerase, which occurs with the help of the sigma factor. Then the local unwinding of the double stranded helix takes place. This local unwinding creates a open binary complex, and then the phosphodiester bond forms, and as soon as the phosphodiester bonds are formed, the sigma factor is released. So binding of the RNA polymerase, unwinding of the DNA, and binding of the RNA polymerase, unwinding of the DNA, and formation of the phosphodiester bonds are the 3 Major steps of transcription initiation. So I hope transcription initiation is clear to you. We have also studied the EcoR1 promoter sequences and all. So I hope this transcription initiation is now clear to everybody. Just a minute. There is some technical glitch, it seems. Tell me, sigma factor is responsible for
Initiation process.
initiation process. Fine. So sigma factor is important for the RNA transcription initiation. So the answer is D. Okay. Next is assembly of the core complex occurs with the help of. We are talking about the core complex. It occurs with the help of alpha subunit, beta dash subunit, beta subunit or gamma subunit. So Aditya, Janvi, Tejaswini, Shivani, Swarna, they all have answered as A, that is alpha subunit. Jehanara, okay, yes, the correct answer is A. Alpha subunit is responsible for the assembly of the core complex. Fine. Now we'll move to transcription elongation. So now we have studied that the up till the formation of first phosphodiester bond, it is your initiation. Okay. After the formation of first phosphodiester bond, the sigma factor is released and the transcription bubble keeps on walking. It's like, suppose if this is your DNA, first here it is transcription bubble, then after some time transcription bubble will come here, then here. So it's like transcription bubble is walking. So rewinding and unwinding continues. Okay. So it looks like transcription bubble is walking and RNA chain continues to grow and RNA polymerase is bound to both DNA template and RNA. This is important. This point is important, that RNA polymerase, it is bound to both the DNA template and it is bound to the RNA. So this is what transcription elongation is. Transcription initiation is a little, there is much more to study about transcription initiation. Transcription elongation is basic, that the unwinding and rewinding continues. It looks like a transcription bubble is walking and RNA chain continues to grow. Okay, this is your transcription elongation. So now important point here is this table. Now this table I have taken, so you can, I'll upload my slides also, you can see from there as well. So you can see whatever image I have taken from the PDF, I have written over here, image taken from lectures. So these whatever screenshots I have taken because they are important. And when I say important, not just important for your NPTEL exam point of view, but in general they are important. Even if you are not giving any competitive examinations, for your semester examinations also these are important. So why it is important? Because sigma factor, there are different types of sigma factors. Okay. So when I say that sigma factor is responsible for the recognition of the promoter sequence, so that means that there is not just one kind of sigma factor which is involved. There are actually many different kinds of sigma factors. So you can see these all are very important. So if you have Pathfinder, you will find it there also. So basically sigma 70 is the one that we consider mainly. It is responsible for housekeeping genes. Sigma 54 is responsible for nitrogen metabolism. Sigma 32 for heat shock proteins,
and sigma 28 for flagellate synthesis and chemotaxis. So this is 28, 28, 32, 54 and 70. So 70 is responsible for housekeeping gene recognition, 54 nitrogen metabolism, 32 heat shock genes, 28 flagellate synthesis and chemotaxis. This is about transcription elongation. Now we'll see about transcription termination. So up till now we have studied that okay, RNA polymerase is coming with the help of sigma factor. It is able to recognize the promoter sequence, specifically -10 and that is Tata at and -35 TTGACA. These are the two sequences where consensus sequences within the promoter which are recognized by the sigma factors. Okay, then what happens? Then the DNA would be converted, it will be melted, transcription bubble will form, and RNA polymerase will form the first phosphodiester bond. After the formation of first phosphodiester bond, sigma factor is released and transcription bubble keeps on walking over the DNA, and in this way transcription continues. RNA polymerase, it is bound both to the DNA template as well as RNA. Okay, now transcription has to stop somewhere also. Now how transcription will stop? It should get certain signals that will tell us that okay, now transcription has happened a lot, now you should stop the doing transcription. So there must be some signals. So those signals are of two types. First are row dependent and the another one is row independent. So which are row dependent? That these are yeah, okay. So the row dependent terminator means that they require a row factor. Okay, so I think just for a minute, I'm a little confused that row dependent would be intrinsic or row independent would be intrinsic. So I'm not very sure about the second point. Okay, so I will clear it up and I will tell you in the next class that row dependent and row independent are whether it is intrinsic or extrinsic. So does anybody has idea that whether what is written is correct or not? If you have any idea, you can tell everybody, you can tell the class. Row dependent is intrinsic or extrinsic? Any idea, anybody? So nobody is answering. Fine. So we'll just clear this thing. I think row dependent is the extrinsic one. This should be extrinsic. That means, and this should be intrinsic. Okay, this is okay. So now termination occurs with the help of row dependent or row independent methods. So row dependent means it requires an external row factor. It requires the involvement of an external row factor, and because there is an external requirement, that is why it is known as an extrinsic method of termination. And so basically what happens that there are so in row dependent, I'll just okay, I'll tell it in my way. So one is row dependent. So in row dependent, you need a row factor. Okay, you need a row factor, and because this row factor is needed, therefore it is not an intrinsic termination, this is an extrinsic termination. And the one which is row independent, okay, The one which is rho independent, it does not require the rho factor. So how then transcription happens? Transcription happens because at the end of the mRNA, between the mRNA and DNA, so there are some inverted loops. So there are loop formation occurs. So in rho independent method, actually loop formation occurs, and this loop formation occurs because of the inverted sequences which are present. Okay, so this loop formation occurs, and then what happens, that between the DNA and the mRNA, AU bonds are formed. AU bonds, and these AU bonds are weak. So what happens, because of this loop keeps on getting increasing. So this loop kinds of puts a weight, and it kinds of pulls down this mRNA, and because the bonds at the end are also AU, so it can easily be pulled off. Did you understand? Here in rho dependent, you need a rho factor, and this rho factor binds at the rut sequence. This rho factor, it binds at the rut sequence. Whereas in the case of rho independent, no rho factor is there, but a hairpin kind of loop is formed, and as this loop keeps on increasing, it kinds of pulls the mRNA, and because the bonds are also AU, it can be easily pulled off. So these are the two methods, rho dependent and rho independent. Now we'll see these two questions. So the question is, rho independent transcription termination require rho independent, we are talking about, which does not require the rho factor, unique DNA secondary structure, unique RNA secondary structure, unique poly A signal, or coupled transcription translation. What is the answer? Tell me. Rho independent. Anybody? Answer whether it is right or not, just tell me what you think about it. Rho independent termination requires what? DNA secondary structure, RNA secondary structure, poly A signal, coupled transcription translation. What does it require? So you are saying A. That means unique DNA structure. So you think that DNA is forming a secondary structure? Hairpin loop is formed. In rho independent, the formation of hairpin loops takes place. And where does this hairpin loops takes place? Tell me. Okay, so some Aditya is saying B, Janara is saying C, Janvi is saying C, Kavita is saying B, Riya is saying B. So we have A also, we have B also in options, and we have C also in options. So the answer would be B. Why? Because we are talking about secondary structures. So RNA forms secondary structures, not DNA is forming secondary structure. So if this is your DNA and this is your RNA, okay, see, this is your DNA template. Just a minute. This is your DNA template, okay, and this is your RNA template. This is getting formed. I'm sorry for my extremely bad drawing. This is the RNA template. Now, now what happens, that some inverted sequences are present here, and these inverted sequences leads to the formation of a loop. Means these two sequences are like complementary and they form a loop. Now there is a loop. Now this is DNA. Here suppose A is present, and this is your RNA. Here U is present. Now this loop, hairpin loop, is creating a drag. It is pulling it, and because the bonds are only two, it can be easily pulled also. So this is what happens. So therefore RNA secondary structure is the correct answer. Okay. Now next question is, which of the following is gene specific? Alpha factor, beta factor, sigma factor or delta factor? Which of the following is gene specific? Tell me. Which of the following subunit is gene specific? Alpha, beta, sigma, delta? Think and try to answer. Okay, so Jahanara has said A. Any other option? Tejaswini A. Okay. Anuya A. Okay. Good. I want more participation from the students. No? No more participation. Trishala A. Okay. Good. Bhavya A. Fine. Okay. So the answer is A. A is the correct answer. So why it is gene specific? We have seen also that there are different kinds of alpha factor, you are saying. Oh, I'm sorry. Okay. Why you think A is the correct answer? Can anybody tell me? Why you think alpha factor is gene specific and not sigma factor is gene specific? Think. I want the explanation. If you are saying A, then what is the explanation? Aditya, Bhavya, Trishala, Tejaswini, Anuya. Anybody give me explanation? Aditya, can you tell me why you have chosen A as the option? Aditya, can you tell me? Okay, you must be smiling and thinking, oh god, ma'am picked me. Anyways, so I want you to answer this question. In the next class, you must answer this question. Otherwise, I'll tell you, but you should try to tell me the logic. One more question I will choose, for which I will not tell the logic and you will only tell me the logic. So it is question number 8th, and so Jahanara Momin, please remind me in the next class that we have to discuss question number 8th. And okay, alpha is used to assemble the coenzyme, yes. So it would be gene specific in that case. So it is the responsibility of Jahanara, if you are coming to the next class, which I would insist you to please come, to remind me that this 8th question number and other one more I will choose, I will see randomly, I will choose. You should tell me the explanation. Not, I will not always tell the explanation. You should try and think of what is the answer. Okay. Next is, so we are now at the lecture 4. So we have 2 more lectures left, and I think then we will have a couple more of questions. Yes. Fine. Termination and RNA modification. So we have already studied that in E. coli, basically coupled transcription takes place. That means transcription and translation, it occurs simultaneously because there is no dedicated nucleus which is present. You can see this is the this is the RNA, this is the mRNA, okay, and no, sorry, this is the DNA template, and these are the RNA molecules to which ribosomes are binded. Okay, so once RNA is bound, one RNA is bound by many ribosomes simultaneously. So what, sorry, this is the RNA template. I'm sorry, this is the RNA template. This is the RNA template. These are the ribosomes which are bound, and this is the protein which is getting formed. So what they are saying is that in prokaryotes, what happened? This is one RNA, okay. In this one RNA, many ribosomes bind simultaneously. So it enhances the rate of production of proteins, and it reduces the degradation chances of the RNA. What does it means? It reduces the degradation chances because RNA, you know, is very unstable. RNA is very prone to degradation. Now what happens that in eukaryotes this RNA is formed inside the nucleus. Okay, and when it comes to the cytoplasm, it is already completely modified, like it has 5 prime cap, it has 3 prime poly A tail, so it is protected, it cannot be degraded that easily. But this in prokaryotes, what happens that RNA transcription and translation both are happening in the cytosol. Because of that, RNA becomes very much prone to degradation. Why? Because cytoplasm has a reducing environment. So because cytoplasm has a reducing environment, therefore RNA becomes very prone to degradation. But because this transcription and translation is happening simultaneously, and because so many ribozymes are already attached to this RNA, it reduces the degradation chances of RNA also. So this coupled transcription and translation has this advantage that it is also in a way helping the RNA to preventing the RNA to degrade. Okay, then you can see, now we will see that what are the different kinds of RNA processing that occurs in the case of eukaryotes. So there are, first thing is that as we have studied that the prokaryotic RNA polymerase and eukaryotic RNA polymerase are different. So prokaryotic RNA polymerase is a smaller RNA polymerase, eukaryotic RNA polymerase is a bigger RNA polymerase, and there are different RNA polymerases. There are 5 different types of RNA polymerase 1, 2, 3, 4, 5, and these are the 3 major modifications. These are the 3 major modifications, okay, that occurs in your pre-mRNA, which is formed just after your transcription. So first is capping. So what is capping? That if this is your pre-RNA, pre-mRNA, so this is your 5 prime end and this is your 3 prime end. So at the 5 prime end, 7-methyl guanosine is added. Okay, so what is the purpose of this 7-methyl guanosine addition? It provides resistance to the 5 prime exonucleases. So now this 7-methyl guanosine, which is bound to the 5 prime region, it kinds of protects the mRNA. So now this mRNA is resistant to the 5 prime exonuclease, and this 7-methyl guanosine addition helps with the is added with the help of mRNA guanylyl transferase enzyme. Then second is poly A tail. So that means at the 3 prime end, polyadenosine is added with the help of the enzyme poly A polymerase. And third one is splicing. So what happens in the splicing? When your pre-mRNA is formed, it has introns also, exons also. It is like this. This is your pre-mRNA. It has introns, exons, introns, exons, introns, exons. So all these introns are removed, and now we have only exon number 1, exon number 2, exon number 3. So basically what is happening? Introns are spliced out, and therefore it is known as splicing. Okay, this is what splicing is about. Now comes 2 more questions. Coupled transcription and translation is not for increasing protein synthesis, facilitating transcription termination, RNA protection, prevent RNA secondary structure formation. So what is, tell me the answer. Coupled transcription translation is not for increasing protein synthesis, facilitating transcription termination, RNA protection, prevent RNA secondary structure formation. What is the answer? Try to answer. Tell me anything, like whatever you are thinking, tell me. I have already explained this point. No one is telling anything? You could not understand? Okay. So Kavita is saying A. It is not for increasing protein synthesis. Okay. Janvi is also saying A and Janara is saying B. Okay. So just like we have discussed that when transcription and translation are coupled, that means transcription and translation are occurring simultaneously. So what happens is that as soon this is your DNA, this is your mRNA, these are the ribosomes which are getting bound, and this is your protein formation. Okay, that means as soon as mRNA is formed, proteins are also formed. So because of this, protein synthesis is getting increased. Why? Because in a single mRNA, many different ribosomes are getting attached. Ribosome number 1, ribosome number 2, ribosome number 3. Because simultaneously in a single mRNA, so many ribosomes are getting attached. Therefore, the rate of protein synthesis is increased. And because all this is happening in the cytosol, which has a reduced environment, which can degrade the RNA, but because ribosomes are attached to it, so RNA is getting protected. And because these ribosomes are attached to it, so RNA cannot form any secondary structure, because RNA is not freely available to form any secondary structure. Therefore, it is preventing RNA secondary structure formation also. The question is, coupled transcription and translation is not for facilitating transcription termination. This is the correct answer. That this coupled transcription translation has no role in termination of the transcription. Okay, is it clear everybody? Okay, now next question is, mRNA capping do not ensure protection, translation, splicing, alternative splicing. What is the correct answer? mRNA capping do not ensure. Tell me. Tejaswini is saying B. Any other options? So the correct answer is D, alternate splicing. Because mRNA capping, okay, it is ensuring protection, it can help in splicing, it has no role in inhibiting translation, because it is at the 5' end, it has no role. But mRNA capping, mRNA capping has no role, it does not ensure alternative splicing, because alternative splicing depends on it is a cell specific feature, that in a particular cell whatever genes has to be expressed, those exons are chosen and those mRNA is formed. Therefore, it has no role in alternative splicing. Then we will just quickly move. So just like I said that in eukaryotes there are different kinds of RNA polymerase. So 5 different kinds of RNA polymerases are there. Polymerase 1, 2, 3, 4 and 5. And for the formation of mRNA, RNA polymerase 2 is important. So in eukaryotes, which RNA polymerase is responsible for the formation of mRNA? RNA polymerase 2. Then you can see that okay, this is a little bit, it is a little complicated, you should understand this, that so we have discussed that how transcription happens in the case of prokaryotes. In the case of eukaryotes, what happens? You can see there are so many different kinds of factors. You can see there are so many transcription factors are there. Transcription factor 2D, 2A, 2B, 2F, 2E, 2H. So there are different kinds of transcription factor. The first transcription factor which binds to the promoter region, that is the Tata binding region, is TF2D. Then TF2A comes, then TF2B comes. TF2B is basically from B you can see TF2B. So B stands for bridge. Okay. So it forms a bridge between the incoming RNA polymerase and already existing molecules. Then TF2F. So F has an enzymatic activity, not much important. TF2H is very important because it has helicase activity. So basically TF2H is important because it helps in the unwinding. So TF2H is important. And then comes the phosphorylation. Now this is very important. TF2, among all the transcription factors that occur during eukaryotic RNA polymerase, so TF2H is most important. Why? So what we are discussing, we are saying that when we are talking about eukaryotic transcription, so prokaryotic transcription requirements are different and eukaryotic RNA transcription requirements are different. So in eukaryotes, RNA transcription requires the use of various transcription factors. Amongst all the transcription factors, transcription factor 2H is very important because it has helicase activity also and it has kinase activity also. Now what is the function of helicase activity? Helicase activity is responsible for the unwinding of the DNA duplex, and kinase activity, what does it do? Kinase activity is responsible for the phosphorylation of the carboxyl terminal domain, and because of this phosphorylation of the carboxyl terminal domain, initiation is completed and elongation takes place. So very important, you should always remember this point, is very important. Okay, that the carboxyl terminal domain, it gets phosphorylated, and when this carboxyl terminal domain gets phosphorylated, then only elongation takes place. And the phosphorylation of this carboxyl terminal domain, it occurs with the help of TF2H, because transcription factor 2H, it has helicase activity also and it has kinase activity also. Okay, now we'll see some questions. Okay, so phosphorylation of the carboxyl terminal domain, it ensures what? Elongation, termination, initiation or acceleration. Just now we have studied about it. Tell me. Phosphorylation of the carboxyl terminal domain ensures elongation, termination, initiation or acceleration. What is the answer? I want answers. B. Any other? Everybody is saying B, and it is the wrong answer. Why? Because I just now told that carboxyl terminal domain, when it gets phosphorylated, then transcription initiation is completed and that it undergoes now elongation. So phosphorylation of the carboxyl terminal domain ensures transcription elongation. I am saying initiation is completed, not the whole transcription is getting completed. When initiation is completed, when first phosphodiester bond is formed, after the formation of first phosphodiester bond, the carboxyl terminal domain of the RNA polymerase, it undergoes phosphorylation. And when the carboxyl terminal domain of the RNA polymerase undergoes phosphorylation, then initiation, the first step, initiation is completed and elongation takes place. Is it clear to everybody? Yes or no? Okay, nobody is okay. Janvi said at least yes, ma'am. Fine. Next, state true or false. Every gene require a regulatory sequence. True or false? Every gene requires a regulatory sequence. Yes, true. Kavita is saying yes. I don't know, you are saying for the answer or what? This question is, every gene requires a regulatory sequence. True. Yes. So every gene needs a regulatory sequence. So this is true. Next question is, TSP is the first base of an RNA. What is the first? What is the start codon? Can anybody tell me?
GUC.
AUG, what is the first amino acid which is added?
Methionine.
Methionine. So methionine, which is coded by, methionine is coded by mostly AUG, but GUC is also correct, but mostly methionine is coded by AUG. So TSP is the first base of RNA, true or false? Tell me, what is TSP? Nobody is answering. So basically, the first base of RNA, which is the transcription start point, so it is true. Why? Because the transcription start point, wherever the transcription is starting, that is actually the first base of RNA, which is methionine. It is a little, the language of this question is a little tricky. You just have to understand that where the transcription is starting, the transcription start point will add the first base of the RNA. Okay, so this is true. Then any nucleotide can form the mRNA 5' cap. True or false? Yes, transcription start point. Janvi is saying correct. Okay, the question is, any nucleotide can form mRNA 5' cap. True or false? Any nucleotide can form 5' cap? Yes or no? Any guess? No. Why? What is the what forms the 5' cap? It is false. The answer is false. Okay, Kavita, you can tell. Yes, correct answer. Kavita, you are saying correct. So this is false. Then tRNA do not undergo post-transcriptional modification. True or false? tRNA do not undergo post-transcriptional modifications. True or false? Does tRNA undergoes any post-transcriptional modifications? Any answer? Okay, so you should always remember, yes, so Janvi is saying false. It is correct. Why? Because tRNA also undergoes some kinds of post-transcriptional modifications we might not have covered as of now, but it's not just mRNA that undergoes post-transcriptional modifications, tRNA also undergoes. So this is a false statement. Okay. Now, next question is, in both prokaryotic and eukaryotic cells, the synthesis of protein chain is initiated with. Synthesis of protein chains is initiated with arginine, methionine, serine or valine. So what is the correct answer?
Methionine.
Methionine is the correct answer. Okay. Okay, next question is, transcription in eukaryotes is initiated when RNA strand is present, RNA polymerase is present, core promoter sequence is present, none of these.
RNA polymerase is present.
Okay, but okay, think more about it. So Janvi is saying C, Tejaswini is saying C. Kavita, you have any query? You can tell me. You have raised your hand. Okay, fine. So Janvi, Tejaswini has said C, and I think Janara, someone has said B also. Someone is saying B. Okay, fine. So the correct answer is C. Why? Because RNA polymerase is there. Okay, but if the promoter sequence is not there, RNA polymerase cannot do anything, because this polymerase has to first go and bind to the promoter sequence and recognize the promoter sequence. Only then the transcription can be initiated. So if your promoter sequence is not there, then your transcription cannot be initiated. So the correct answer is C. Okay, next question is, name the site where upstream sequences are located. So upstream sequences are prior to the start point, after the start point, right border of DNA, in the middle of DNA. What is the answer? Tell me. Upstream sequences are, where are upstream sequences present? So Kavita has told A. Janvi has also said A. Okay. Janvi has said B, which is after the start point. Okay. Jahanara is also saying B. Okay. Any other answers? Kavita is saying D, which is in the middle of the DNA. Okay. Fine. So another is there. Okay. See. Start point is given as 1. This is the transcription start point. This is the scale. This region, which is before the start point, is known as the upstream region. This is the region which is after the start point, is known as downstream region. So upstream location sequences are present before the start point, not downstream. Now just to understand this, so I don't know how many of you are studying, or you might have studied about bioprocess technology. So in bioprocess technology we have something which is known as downstream processing. Okay, so downstream processing occurs after our product is formed, and before the product, whatever things we do, that is your upstream processing. So downstream means, down means after. So when we say after, it comes here also with the same analogy. It should come after the start site. So upstream means, up means above. That means it is above, means before the start site. Down means after. So it is after the start site. Like this you can remember. You can also take the advantage of that bioprocess analogy that I am saying, downstream processing. Downstream processing is something which occurs after your product is formed. Okay, then which of the following is true for RNA polymerase activity? RNA polymerase, DNA dependent DNA synthesis, direct repair, DNA dependent RNA synthesis, or RNA dependent RNA synthesis? Tell me the answer, and then I will ask another question from this same. What is the answer? Which of the following is true for RNA polymerase activity? DNA dependent DNA synthesis, direct repair, DNA dependent RNA synthesis, RNA dependent RNA synthesis. What is the correct answer? C. Okay, Tejaswini, Jahanara, you all are. So C is the correct answer. Why? And one important point is, here we are talking about RNA polymerase. Now because we are studying transcription, so we are saying okay, it is C. But in general, we have to, this question is a little incomplete, because RNA polymerase can be used for transcription also and RNA replication also. So if it is for transcription, then it is a DNA dependent RNA synthesis. But if it is an RNA replication, then it is an RNA dependent RNA synthesis. So in this way, this question is a little incomplete. Anyway, now next question is, name one intrinsic terminator of transcription. Intrinsic terminator of transcription. How intrinsic termination of transcription happens? What is the answer of this question? Tell me quickly. Intrinsic. Intrinsic transcription is row independent, because row is a protein. It is a protein which is an external factor which comes and which helps in transcription, whereas intrinsic means that RNA does not need any external agent to terminate its transcription. RNA, with the help of its own hairpin structure, it will terminate transcription. Intrinsic means with the properties itself which are present in the transcription. Within the RNA there are some properties that helps in the termination of transcription. So it does not require the external environment of a raw protein. Okay, so the answer is B. Now we come to the next question. Name the sigma factor which is used for promoter recognition. Which sigma factor is used for promoter recognition here? Mostly which sigma factor is used for promoter recognition? Tell me the answer. Sigma 32, sigma 70, sigma 60, sigma 40. Which sigma factor is used for promoter recognition? No one is answering? B. The correct answer is sigma 70. Okay. Then comes the next question. Okay, these are the last two questions. Sigma 32. So why you think sigma 32? Sigma 7. Jahanara, can you tell me the reason why you chose 32 and not 70? Okay, so question number 18th and question number okay, this question number we will discuss why sigma 70 and not sigma 32, sigma 60 or 40. Okay, we'll discuss it in the next class. It is the responsibility of Jahanara, your only, to remind me. And I'm sorry if I'm spelling your name incorrectly. If I'm spelling it incorrectly, you can tell me how to spell it. Okay. Which of the following codons is the mRNA start codon that initiates translation? Which codon is responsible for the initiation of translation? UGA, UAG, UAA, AUG. 19th question. Aditya is saying AUG. Okay. Any other answer? Any other C? That is AUG. Okay. The correct answer is AUG. So what are these codons? UGA, UAG and UAA. Can you tell me what are these codons? These codons are stop codons. Aditya, you are right. So these codons, Kavita is also saying correctly. Janvi is also saying correctly. So these codons are stop codons. UAA, UAG and UGA. These are stop codons. Now the last question is, which of the following is not dependent on the C-terminal domain of polymerase? Transcription termination, this is the A option, sorry. Transcription termination, splicing, capping, polyadenylation. Which of the following is not dependent on the C-terminal domain of RNA polymerase? 2. Termination, splicing, capping, polyadenylation. Can you tell me? What should be the answer? No? Okay. I'll tell you just one trick. The answer should be A. Why? Because if you see mRNA splicing, 5' capping and polyadenylation, all the three are what? These are RNA post-transcriptional RNA processing events, RNA editing events, splicing, capping, adenylation. Here the only option which is odd one out is transcription termination. So if I choose splicing, then capping and polyadenylation, these are also processes which contributes to the same thing. So here the answer should be transcription termination because it is like the odd one. So always remember, even when you're giving some even if you are going for any competitive examinations, if you don't know anything about the options, try to find the odd one out, which is not going anywhere, or try to find the relation between the options. If some options have some relation, other option has no relation at all, then that can be the possible answer. Here these three are related to each other, as these all the three are the post transcription modifications. One thing, second thing is that we have also seen that the carboxyl terminal domain of the RNA polymerase, this has to be phosphorylated, and when it gets phosphorylated, then transcription elongation takes place. Okay, so it has no role in termination of transcription. It elongates the transcription, and when the transcription is completed, then whatever whatever post transcription modifications, it helps in that. So it has no role in termination, it helps in elongation. So like this you can try to find the odd one out, and you can do it. So I think we have completed the questions. Two questions we will discuss. I want you to answer them, so if you can answer them, it's fine. So with this we have completed. So if you have any questions, you can ask me. Anybody has any questions, any topic that you could not understand, please tell me if you have any queries, any point, any topic that you are not able to understand. So was this presentation helpful? Were you able to learn something new? And would you like the same way, that dealing the theory and answering the questions, or you want just the questions? You tell me. Is this way okay for you guys? Theory plus questions, or you want only questions, or only theory is not possible? So theory plus questions is okay, or only questions you want? Okay, so I will go it like this only, theory and some questions, theory and some questions. Okay, okay, fine. So I will go like this only. So this time I have made so if you I will share this PPT with you in 2 or 3 maybe 2-3 days. Okay, and so you can go through it, because if you cannot find the recorded PDF, then I think with this slide I have included the screenshots of the important points which are in the live in the videos. From the videos only I have paused, and I have taken the screenshots, and I have added in the PPT things which are important. If the PDFs are not available, you can go through it. Okay, so with this we end the class. I hope you could get something new, and you could learn this RNA transcription with a more wider perspective and a more wider knowledge and logical and reasoning skills. And if you have any problem, you can always come to me, and you can always ask in the live sessions any problems related to RNA biology, or any problem in general related to molecular biology or science. So with this we end the session. So bye everybody, have a great week.